Hello everyone. Today we're going to be talking about chromosomal abnormalities. So, meiosis is a precise process, but errors can occur, and when they do, they result in chromosomal abnormalities that have significant consequence on the offspring. So let's look at errors that can occur during crossing over in meiosis. So let's look at this first diagram. During prophase 1 of meiosis, homologous chromosomes pair up and exchange segments through crossing over. If this process is imprecise, several types of chromosomal mutations can result. So you can have insertion, which is this one here. And what happens in insertion is a segment of the DNA from one chromosome is inserted into a non-homologous chromosome. And we can also have deletion here. Now, that is when a segment of a chromosome is lost entirely during the exchange. We have duplication, which refers to the segment being copied so that it appears more than once on the same chromosome. And there's also inversion, which is where a segment is reattached in the reverse orientation. The sequence of genes is flipped. That's inversion. Now there's also translocation. Now, translocation occurs when a segment of one chromosome breaks off and attaches to a non-homologous chromosome. Unlike insertion, translocation involves a mutual or one-way exchange between non-homologous chromosomes. So translocation doesn't necessarily change the total amount of genetic material, but it disrupts gene arrangement, which can affect gene expression and produce abnormal phenotypes. There's also something else called aneuploidy. So aneuploidy refers to an abnormal number of chromosomes in a cell. That results from errors in chromosome separation during meiosis. This error is called non-disjunction, where homologous chromosomes or sister chromatids fail to separate correctly. Now, if non-disjunction occurs during meiosis 1, both chromosomes of a homologous pair end up in the same cell. If it occurs during meiosis 2, sister chromatids fail to separate. The resulting gametes have either one extra chromosome or one fewer chromosome. When fertilization occurs, the zygote has an abnormal chromosome number. So a well-known example of this is trisomy 21 or Down syndrome. So that's where we have 3 copies of chromosome 21. So you can see here, got 3 copies, and they are present due to non-disjunction. So that's an example of aneuploidy. Okay, so to summarize, errors during crossing over in meiosis can produce insertions, deletions, duplications, and inversions, all involving changes to chromosome segment arrangement. Translocation involves segments moving between non-homologous chromosomes. Aneuploidy results from non-disjunction, which is the failure of chromosomes to separate correctly, which produces gametes and offspring with an abnormal chromosome number such as trisomy or monosomy. Follow for daily QCAA content.